Good morning, everybody. I would like to welcome you at our presentation of Physiological Days at the Institute of Physiology. My name is Jiří Paleček. I'm the Deputy Director of the Institute. So welcome again. And today I would like to give you a very short overview of what the Institute of Physiology is doing, what our departments are interested in, what we want to deal with, and hopefully strike our interest about it. And then will come the representatives of individual laboratories, which will give you a more detailed overview of what they are doing. So I think that for you sitting in the room, you know where we are. But for those online, maybe it will be interesting to know that we are in Prague, which is located in the middle of Europe, in the Central Europe. So it's easy to travel to other cities in Europe. And we are located also close to the center of the city, which takes about half an hour by public transport. We are located in the campus of the Krch Biomedical Institute, whereas some other institute like Institute of Molecular Biology, Institute of Experimental Medicine, and which is nice campus because we can collaborate with the other institutes and use the center facility together. Maybe you didn't know that our institute has a long history. We celebrated 70 years anniversary last year, and it was founded in 1954 by medical doctors. So at that time, these gentlemen, which are noted here on the slide, they were medical doctors, and they were really interested in how the diseases, what are the mechanisms, and how they could help their patients. So they started this Institute of Physiology, and the topics were generally neurophysiology, muscle regeneration, heart adaptation, and ontogenesis. And throughout the 70 years, the topics developed, and today we are generally concentrating on the neurophysiology, cardiovascular physiology, and metabolism. So these are the major topics that we study at this institute, and we are really trying to put them together. So we want to look how the civilization diseases like diabetes, Alzheimer's disease, and other develop, what are the mechanisms and how we could improve the life of patients to try to find some new treatments for these diseases. And for this, we use different mechanisms, different models, and we are arranging the models from molecules up to the patient cohorts, trying to figure out how these diseases develop. So the institute itself has about 23 research laboratories. The number is changing because we are establishing new laboratories with new young principal investigators. We are about 71 principal investigators at the institute, which means these are funded by their own grants from different grant agencies. And as you can see, there are also a number of PhD students and, of course, also undergraduate students like bachelor and other students. When you would look at the webpage of the institute, you could see the details of each of the departments. And in the neurophysiology part or neuroscience part, you can see we are ranging from looking for the function of different molecular ion channels and biological rhythms, neurochemical, to different cholinergic signaling, to some diseases like epilepsy, memory, Alzheimer's disease, and pain research. In the cardiology part, we have a very long tradition department of development cardiology, experimental hypertension. We have also a department which is really concentrated on developing models of animal models of hypertension. We have also quite very good department of biomaterials and tissue engineering and biomathematics, which is helping us. And in the area of metabolism, there are a number of laboratories looking at the adipose tissue metabolism, bioactive lipids, and also mitochondrial diseases and the function of mitochondria under different pathological conditions. So all the details, if the laboratories are not presenting today, are available on the website and you can go into it. So why is it important? I think I already mentioned that the increase of metabolic diseases and obesity is increasing as the population is also getting older. The number of Alzheimer's disease patients is increasing, and the coronary heart disease and myocardial infarction may be in fact decrease the mortality, but it's still the leading cause of mortality in the patients in the older population. So all these diseases are very important to study, and in most of them, we are really looking for new mechanisms and how to treat them, how to help the patients, and how to deal with these civilization diseases. So what do we do? As I noted before, we are using different types of approaches from the molecular level, in vitro systems, models on laboratory rodents, up to clinical cohorts that we study using different omics, possible methodologies like genomics, transcriptomics, etc. But each lab has its specific methodology. So you can hear about it today. You can also visit the laboratories in the afternoon and see what specific methods they are using, which are highly interesting. The mechanisms we are studying is ranging also from inflammation, neuroinflammation, energy metabolism, neurohormonal regulation, function of different receptors, their pharmacological changes, and higher nervous function. So all this is studied, and as I said, we have specialized methods in each department, but we have also some core facilities. We have very nice microscopic facility where we have a number of different optical methods. We have proteomics facility, metabolomics facility, and as I said, there are also some facilities within the campus and also in BioCEF. I don't know if you heard about BioCEF. We have some laboratories in BioCEF as well, so we are part of that consortium, and we can use these facilities as well. And not least but last, we have also a nice animal facility where we have a number of animal models, transgenic mice, and specialized rat strains where we can study specific changes during some pathological diseases. The work that I was talking about is supported by grants. We have a number of grants from Czech sources. We have also grants from international sources. And for the last 5 years, we were part of the Czech Recovery Plan, the Next Generation EU, where we had 2 huge grants. One is called Cardia, where we were studying the metabolic and cardiovascular diseases, and also we were part of the National Institute of Neurological Research. So these are the supportive money we are getting for our research. And we have also wide collaborations within the Czech Republic. I think one of the most important is the Institute of Organic Chemistry and Biochemistry, which in cooperation, we are developing some new drug treatments. Also the Institute of Clinical and Experimental Medicine, which we collaborate on the patient's part. We have a number of other collaborations by the individual scientists and principal investigators. And of course, we have also some international collaborations, which I think not complete here, but there exist. So this is about the Institute in very brief, short presentation. And why we need you? Well, we need you so we can help you. You can help us to work and find new ways how to define the mechanisms in the diseases I was talking about. So we are looking for really enthusiastic, motivated students, either bachelor or a diploma thesis, which then can become a PhD students, or PhD students coming from abroad or from Czech Republic or from different faculties. And I think about the program for the PhD students, Olga will tell you more about now. So thank you for attention now.
Well, thank you, Jirka, for the introduction. My name is Olga Zimmermanova, and I am responsible for all research activities, and one of them is this Day of Physiology and Medicine for you from universities or from abroad. Because we need, as Jirka said, we need motivated students, and you can see here the list of topics of that students are involved in at our institute. That is a broad range from analytical chemistry, animal physiology, up to the neuroscience. And the most popular are animal physiology, biochemistry and pathobiochemistry, and molecular and cell biology and neuroscience. Our students are always students also at some university in the Czech Republic or at some abroad at some institution. And during last 5 years, more than 60 60 defended theses were from bachelor's, master science students, and about almost 60 of PhD students. And currently we have opened a PhD call for PhD students, which is online, the application. You can register via this QR code, and the deadline is March 15. So you can study here the projects in neuroscience, cardiovascular physiology, or metabolism. And there is a list of some benefits that we offer for students, such as Jirka mentioned, there is a broad range of methods from physiological field, biochemical or molecular biology field. And our students are usually employed. It can be up to full-time salary, depending on the laboratory where they study. And we, as you will hear at the end of this morning session, we organize regular events for PhD students. There is, in the third year of your PhD study, there will be an advancement report, which is helping you to proceed smoothly your PhD study. We also have an internal PhD thesis evaluation that can help improve your final PhD defense at the university. We offer also English language courses, and in addition, for students that are not from Prague, we offer also on-site accommodation, and there is a nice campus full of birds, full of animals, and also some sports facilities. And as concern bachelor and master science program, at this part of the web page of our institute, there is a list of available topics, and some of them you will hear also during the morning session. And I recommend here to contact particular supervisors that write them an email, or today you can speak with them in the afternoon session. So this is our morning session, the program, and afternoon, after lunch, you will have the possibility to visit up to 3 laboratories. You will please register for the visits in the particular laboratory that you select. In the foyer there is a list, and the visits will start at 1:45 and 2:30. And I think this is the end of the introduction, and I wish you to enjoy this day, and we can continue with the presentations. Although we are a little bit ahead of the program, but I think it doesn't matter. We can start with the presentation. As the first speaker, I will ask again Jiří Palecek because he's a head of the laboratory of pain research, and he will present the topics.
Thank you. So I am back. So I'm taking you to the world of pain. I hope it won't be a painful experience for you. But what we are really trying to do is to understand the mechanisms of pain and find some possible new targets for new therapies. So when I would ask you what is pain, maybe you would answer me something, but we don't really have time for questions and answers at this topic now. But you probably know that there is acute pain and chronic pain, and while the acute pain is important for you, for your survival, to know that something damaging is happening to you, the chronic pain is really a pathological state with high impact on the patients. And of course, there are different types of pain, and one of those very difficult to treat are neuropathic pain states. Neuropathic pain comes from damage of the nervous system. It can be peripheral or central nervous system. You can see there are a number of pathological states which can lead to neuropathic pain. And in fact, what is important to note is that the mechanisms of neuropathic pain are different from the mechanisms of normal pain, the pain that you feel when you burn your finger or hurt your leg or something. And these mechanisms are different, so the classical analgesic drugs don't work on neuropathic pain, and that is why it's important to study these mechanisms and find new treatments for the patients to alleviate their pain. So we really need new analgesic drugs with minimal side effects that will be effective not just for the acute pain, but also for the chronic neuropathic pain states. When we talk about how to modulate pain, we can look about the pain pathway, and we would talk about all the differences and details, but we are really concentrated on the modulation of nociceptive transmission at the spinal cord level, and that's what is really interesting for us. We are using different models of neuropathic, inflammatory, arthritic pain, and looking for the role of neuroinflammation, cytokines, chemokines, different receptors like TRPV1 receptors, which are activated by capsaicin, opioid receptors, and cannabinoids in the pathology and modulation, and especially in the mechanisms that lead to developing these pathological pain states. We have several collaborations, like on the diabetic neuropathy with the Institute of Clinical Experimental Medicine, where we study cohorts of patients. We have a grant project on chemotherapy-induced neuropathic pain with Texas University, and on the role of interleukin-4 in the mechanisms of peripheral neuropathy development with the University in Innsbruck. We have also a very nice collaborative project with the Institute of Organic Chemistry and Biochemistry, where we are developing new neurosteroid drugs for treatment of these pathological pain conditions. The methods we are using, of course, it ranges also widely, but our major method is electrophysiology, especially electrophysiology recording, patch clamp reporting from single cell neurons. I don't know if you know what is patch clamp. Probably not. Maybe you will learn about it in the afternoon if you will join our department. You can see an example of that. Here is a spinal cord slice from rat. You can see the recording electrode here in more detail attached to the cell, and we can record excitatory currents, synaptic excitatory currents at this neuron, and here they are evoked by capsaicin application, which activates TRPV1 receptors, leads to glutamate release, and which we can register as postsynaptic excitatory currents. We are also using optogenetics, optical stimulation of a specific population of neurons, calcium imaging in neuron cultures, behavioral tests, and other immunohistochemical and chemical methods which are available to study these mechanisms. Just to give you a hint about what we are doing, here's an example of one project on chemotherapy-induced neuropathic pain. Maybe you know, maybe not, that a number of the cancer treatments like paclitaxel, bortezomib, cisplatin is leading to neuropathic pain states in the patients, and it's very difficult to treat those pain states. So in fact, some of the patients will stop the chemotherapy treatment because of the pain they are experiencing. So it's really a very serious side effect, and there are no good treatments. So we went to study how this chemotherapy-induced neuropathic pain is developing. And as I said, we are mostly interested in the changes at the spinal cord dorsal horn, whereas the synaptic transmission between the primary efferents conveying the nociceptive inputs to the postsynaptic neurons. And in this paper, I would like to demonstrate some of our methods. So we are using the behavioral methods, and you can see that in this model of paclitaxel-induced neuropathic pain, we induced increased mechanical sensitivity in the animals which went through this paclitaxel treatment. However, here we tested duvelisib, which is a drug, which is a phosphoinositide 3 kinase specific inhibitor. What is important about this drug is that it was approved by FDA for treatment of some hematological diseases. But as it is a specific antagonist, we wanted to try or test it in this model because we showed in previous papers that the inositol phosphoinositide 3 kinase is important for the development of the neuropathic pain. And as you can see, really, with increasing the dose, we can alleviate the mechanical allodynia, the increased mechanical sensitivity in these animals. And we have also shown that the chemotherapy leads to increased sensitivity of TRPV1 receptors. You can see that the activation of dorsal horn neurons in spinal cord When we apply capsaicin under normal conditions, you see a nice increase in activity. However, after induction of the model of paclitaxel, the increase of activity is highly increased. You can see it also here on the graph. These are the control responses, the blue one with the paclitaxel, the pathological one. And with treatment with the duvelisib, with the specific antagonist, we could decrease this highly pathological activity as well. We were also using in these experiments the optogenetics. We were applying short 5 millisecond blue light pulses to our neurons, and you can see we evoked nice inhibitory currents in excitatory neurons by the slight stimulation and activation of channel rhopsin in the neurons. And again, there was a decrease of inhibitory currents, which is known to be happening in the pathological state. So there is a disbalance between the excitation, which is increased in pathology, and decreased of inhibitory moments, which leads to enhanced nociception and pathological pain. And again, you can see there were some good changes with the treatment. We did also immunohistochemistry analysis of those root ganglion neurons. These are the neurons which connect by the primary efferents the periphery with the spinal cord. And we have shown that the changes in the phosphoinositol 3 kinase activation are changed with the duvelisib treatment. And as one last example of our work, we are doing now the diabetic neuropathic patients. We are studying changes in their plasma levels of different metabolites and lipids and also exosomes. So here is also a nice picture from human plasma where you can see exosomes in the plasma together with some extracellular vesicles which are not exosomes because they don't have the plasmatic membrane. And we can do metabolomic analysis of these samples as well, looking for the active molecules that could be involved in the development of this pathological pain state. And that's about all from me. We are really looking for some motivated pre-graduate students, also PhD students, and you are welcomed to join us, and you can talk to my students and collaborative workers in the afternoon. I won't be there present, but I'm sure they will answer oral questions if you have any. Thank you.
Thank you, Jirka. And our next presentation will be from the Laboratory of Neurochemistry laboratory.
Good morning, everybody. My name is Janek Kubik. I'm from Laboratory of Neurochemistry. And in my much shorter talk than my previous speaker, I will give you some broad description of things we do, and it can be covered by mechanism of selective modulation of muscarinic receptors. And we are able to accommodate students in each level of their career, meaning bachelor, master, and PhD students. So what are the muscarinic receptors? Muscarinic receptors are metabotropic receptors for acetylcholine, and they fulfill a wide variety of physiological functions. Here we have their role in peripheral nervous system. Probably, as you know, acetylcholine is a major neurotransmitter of parasympathetic neurons in periphery. And on the innervated tissues, it acts via muscarinic receptors that they mediate a wide variety of functions like bronchoconstriction or of a smooth muscle via M2, M3 receptors, or they mediate heart. They are causing bradycardia and also have a cardioprotective effects. In pancreas, where M3 receptors, they stimulate insulin secretion, so they have a quite important role in type 2 diabetes. And in intestines, via M2, M3 receptors in smooth muscle cells, so they mediate motility of the of the intestine, so they actually counteract against the opioid constipations due to medication. And they are also present in smooth muscles, in constrictors and detrusors on bladder. So antagonists can be used for incontinence or increase on overactive bladder. Also, muscarinic receptors mediate a wide range of functions in the central nervous system. Here we see that the expression of individual subtypes in brain, and we see their participation in functions and associated diseases. So, for example, M1 receptor we will find mainly in cortex, brain cortex, and some subcortical ganglia, and it mediates cognitive functions, and its impairment is associated with Alzheimer's disease or Parkinson's disease, or if the problem is in hypothalamus, then it's associated with epilepsy. M2 receptors you will find in a similar location as M1 receptors, but they are not located postsynaptically but presynaptically, and are associated and are mediating autoinhibition of acetylcholine release, and some disbalance in M2 signaling leads to epilepsy, or some M2 receptors in dorsal root ganglia, they mediate analgesia. So things that you heard about in previous presentation. M3 receptors are involved in cognition, long-term depression, and again, their imbalance is associated with epilepsy. M4 receptors are mainly expressed in subcortical ganglia, and especially in striatum, and they are mediating some positive and negative effects of schizophrenia. They are also involved in epilepsy and analgesia. And M5 receptors, they have very limited expression to the ventral tegmental area, that is in a so-called reverse circle with dopamine neurons, and that they're involved in addiction. And there is also some expression in of M5 receptor in microvasculature, so they mediate supply of glucose and oxygen to the brain. So as you can see, the muscarinic receptors are involved in many functions and associated with many diseases. So they become a quite good pharmacological target. So if we can somehow negatively modulate, either by antagonists or negative allosteric modulators, M1 receptors, we can treat Parkinson's disease or epilepsy. M2 and M3 antagonists are good for chronic pulmonary obstruction disease or irritated or active bladders and incontinence. M4 receptors again treat Parkinson's disease, and M5 are theorized to prevent withdrawal symptoms and development and addiction, are also to block schizophrenia psychotic symptoms. On the other hand, when we try to stimulate or positively modulate these receptors via agonists or positive allosteric modulators. So if we positively modulate M1 receptors, we can treat Alzheimer's disease or cognitive deficit in schizophrenia. With M2 and M4 receptors, we can have analgesic effects. M2 only centrally active may be good for Alzheimer's disease. M3 agonists may be good for type 2 diabetes, and they are used to treat dry mouth symptom. M4 receptor, besides the analgesia, are good to treat the psychosis symptom in schizophrenia, and M5 receptors, activation of M5 receptors have procognitive effects in various forms of dementia, and that's why we said that they are expressed in microvasculature in brain, and their activation supplies brain with more oxygen and glucose upon activation. So if you look at this, it's really nice that we have a quite huge pharmacotherapeutic potential, but the drawback is that we have to be really precise in modulation of these receptors, that the effects of drug should be limited to single subtype or even tissue specific. So it's made a challenge, because if you look at these pictures that are cross-section of crystallographic structures of 4 muscarinic receptors, with also electrostatic potential on them, that positive is in blue, electronegative is in red. And in the ovals, the ovals indicate orthosteric binding sites where neurotransmitter acetylcholine is bound. So you see that these binding sites are virtually identical. The charge is also identical. The shape is identical. So it's really, or virtually impossible to get ligands that would be selective based on the affinity for the orthosteric binding site. So how we can solve this problem? Here we have a structure of M2 receptor with antagonist quinoclinium benzylate, and this antagonist is non-selective. Here we have a structure of antagonist AFDX384, and what we see, that is selective for M2 receptors, and what we see that it has some part of the molecule that goes out of the orthosteric binding site and interacts with the amino acid residue that are not conserved and gains the selectivity. So the way is how to get the selective antagonist is make them bigger so they reach the ectopic site of the receptor. Then here is the structure of M2 receptors with agonist hyperoxo. And what you can see is here is that upon the activation of the receptor, the orthosteric binding site is closed by tyrosine lit. So we cannot, like in antagonist, we cannot somehow grow agonist to reach out of the orthosteric binding site to gain some selectivity. But what we can do is develop allosteric modulator. Here we have an allosteric modulator bound together with hyperoxo. And these compounds are binding to the extracellular part of the receptor that is not conserved among receptors, and their action are selective. And these allosteric modulators can be positive, like in this case, it means they increase the affinity and efficacy of eperoxo or also acetylcholine, or maybe negative ones that they negatively modulate activity of acetylcholine. That is good, for example, if you have a condition with overactivated muscarinic receptors, like in asthma. So yes, so that's all for me, and thank you for attention.
Thank you, Jan. And next speaker is Helena Jančková from the quite new laboratory, recently established, of cholinergic signaling.
Thank you. So, good morning. My name is Helena Jančková, and I am the head of the Laboratory of Cholinergic Signaling. As you have heard, we are a new laboratory, and we are quickly growing. So the people you can see here is actually now only a smaller part of the laboratory, although it's quite dynamic. So people sometimes are asking me what a cholinergic signaling actually is. I believe that most of you know what cholinergic signaling means. No? Okay. Then it's good I implemented this slide. So cholinergic signaling is based on the neurotransmitter acetylcholine that is being synthesized in the presynaptic neuron, and it is released in the synaptic cleft. And after it is released, it binds to acetylcholine receptors, either muscarinic or nicotinic acetylcholine receptors, and it activates them. So the primary mission of our lab is to understand what are the consequences of the activation of the acetylcholine receptors. These consequences, we can observe them at multiple levels. They can be observed at cellular level, when we can see changes that occur in the neuron that was just active, or which receptors were just activated. We can also observe consequences at the neuronal network level. So the neuronal networks, the local network or global network, can be activated, or the signaling can be changed by the activation of acetylcholine receptors. But we can also observe effects on the level of organism. So the behavior or cognitive functions of the whole organism can be changed based on the activation of these receptors. And today I will mostly focus briefly on this most complex level of consequences on the behavioral changes. So how do we study them? I mean, you can imagine we use drugs that activate acetylcholine receptors. But the problem is what I'm trying to illustrate in this slide, that the acetylcholine receptors, either the nicotinic ones as in this example, or also muscarinic, are very widely expressed throughout the mammalian brain. So they are virtually everywhere in each brain structure and also in many neuronal types. And the function of these receptors in each particular place may be different, even antagonistic. So our mission, we try to modulate the acetylcholine receptors in specific neuronal types and in specific brain structure to achieve a more specific effect that is better controlled and is more predictable. So I would say if we should summarize, what we want to do is to determine the localization and function of acetylcholine receptors in specific neuronal types, and to understand, to learn how we can modulate them more specifically. And I will show you today one very brief example of this mission. So here what you can see is the expression of different subtypes of nicotinic receptors in various neuronal types in the prefrontal cortex. And you can probably appreciate this expression pattern is very complex. So this was derived from literature. This is what's known, more or less, about the expression of different nicotinic subtypes in the prefrontal cortex. And to disentangle this complexity, we wanted to focus on a single nicotinic subunit, and we are focusing on beta-2 nicotinic subunit in this case, which is one of the most commonly expressed nicotinic subunit. And we wanted to modulate this subunit in a particular neuronal type in the prefrontal cortex and observe whether we can achieve with this modulation any changes in behavior of mice. So first, we wanted to understand the expression of the beta-2 nicotinic subunit in the prefrontal cortex. So we used fluorescence in situ hybridization. And here in this example, you can see in red color, the red signal is signifying the presence of beta-2 nicotinic subunit, and the green signal is signifying the presence of vesicular glutamate transporter 1, which is a marker for excitatory neuronal types in the prefrontal cortex. And overall, we observed that the beta-2 nicotinic subunit is expressed in both excitatory and inhibitory neurons in the prefrontal cortex, although the extent of the expression is higher in the inhibitory neurons, where it reaches about 80% of neurons are positive for the beta-2 nicotinic subunit. So we were focusing on inhibitory neurons in the prefrontal cortex, and we digged a bit deeper. We checked the expression in some of the particular neuronal types of inhibitory neurons in the prefrontal cortex, and you can appreciate here that all of these examples do express to some extent beta-2 nicotinic subunit, but in the end we decided to focus on NPY, neuropeptide Y, positive or expressing inhibitory neurons in the prefrontal cortex, and we wanted to induce knockdown or decrease of function of beta-2 nicotinic subunit in these neurons and observe whether we can see any changes in mice. So to do this, we need to be quite specific with our knockdown. So because the knockdown has to be regional specific, it only can affect prefrontal cortex, and it has to be also cell type specific because it only can affect neuropeptide Y positive neurons. So for this, we chose to use CRISPR approach, and we crossed mice that do express Cas9 enzyme in a Cre-dependent manner. We crossed these mice with an NPY Cre mice, which are the mice expressing Cre recombinase in neuropeptide Y positive neurons. So the offspring of this crossing is expressing Cas9 enzyme only in neurons expressing neuropeptide Y. And this offspring was then injected with AAV virus expressing guide RNA that is targeting the gene for beta-2 nicotinic subunit. And then only the cells expressing both Cas9 and the guide RNA against beta-2 were susceptible to the knockdown of the receptor. So these mice, in the end, we subjected to a whole battery of behavioral tasks. We mostly focused on functions and behavior that is traditionally associated with the function of the prefrontal cortex, such as locomotor activity, social behavior, attention, working memory, and so on. But today I am going to the end. I don't have much time in this presentation, so I will just show you one single example of social behavior. So social behavior or social interaction in mice can be studied with a simple task. All of them are more or less variation of a task where a tested mouse is let freely interact with either another mouse, which we call social stimulus, or it can also interact with just an empty container or just an object, which we call non-social stimulus. And you can see at first in this graph that regarding this interaction with either object or the mouse, we did not see any changes at first, any differences between the mice that were injected guide RNA targeting beta-2 subunit and that were injected with just scrambled guide RNA, which is a control in our case. So we were wondering why is that, because we really wanted to see something, and then we were thinking maybe the CRISPR knockdown is quite heterogeneous, and we saw it in the brains of our mice that the expression also of the virus was quite variable. So maybe that is why. And for that purpose, for this reason, we correlated the behavior of the animals with the number of cells that were successfully transduced with the virus and that also expressed Cas9. So they were double positive, and only these cells could carry this knockdown of the beta-2 subunit. And you can see on the right graph that we can see a positive correlation between the number of successfully transduced cells, Cas9 positive cells, and the interaction time, the time the mouse spent interacting with social stimulus. So we can see the social behavior, the social interaction is correlated with the number of, or with the expression level of beta-2 nicotinic subunit in NPY positive neurons in the prefrontal cortex. On the other hand, we don't see any correlation in interaction with the object, or we don't see also any correlation in control animals that were injected with the guide RNA, so that was scrambled. So yeah, this is the conclusion I just said. The decrease in the expression of beta-2 subunit is positively correlated with the decrease in social behavior. So with this, I would like to thank you for your attention. And if you have further questions, please come in the afternoon to see our lab.
Thank you, Helena. And our next presentation will be done by Elena Filova from the Laboratory of Biomaterials and Tissue Engineering. And she will present two topics. Yeah.
Good morning, ladies and gentlemen. My name is Elena Filova, and I work in the Laboratory of Biomaterials and Tissue Engineering. And the head is Professor Lucia Bačáková. We have a laboratory here and one small also in BioSef. So we are working in the field of tissue engineering. It is an interdisciplinary field. When we need some biomaterial that is prepared by engineers, chemists, or physicists, and then we construct or try to prepare artificial tissues, for example, vessels, bones, cartilage, skin, etc. So we work mostly with cells, and we study this interaction with material. There are some examples. The three main streams are cardiovascular tissue engineering, bone tissue engineering, and skin tissue engineering. In a cardiovascular engineering, we want to mimic the structure of vessels. There is tunica intima, tunica media, they are most important, and also tunica adventitia, and we want to prepare prostheses that contain endothelial layer and the layer of smooth muscle cells like in vessel. Here is one approach using some gel systems. There is collagen gel and nanofibers. We see cells or nanofibers when they grow, they start to migrate into gel, and cells can be mixed with gel as well. They create some capillary structures, and they migrate to the top of the gel and create an endothelial layer. Or we can use some other systems as well. For example, decellularized tissues, or also some artificial sheets of polymers as well. So we use also both static and dynamic culture, many times of stem cells and also primary cell cultures. And we have also some bioreactors for dynamic stimulation. This supports differentiation of stem cells. For skin, there is like very simple model. So we want to also mimic this layer of epidermis and dermis, where we use keratinocytes and also fibroblasts, some other stem cells for adipose tissue derived, for example, for patients. Here is some construct that contains like simulated layers that can be used as a skin or dermoepidermal skin construct. Again, nanofibers, some gels. There are more options with gel to use. And culture of cells, it's better to have it longer. And we combine cells like fibroblasts, ASCs, Wharton jelly stem cells, and seed keratinocytes on the top. And this construct is similar to skin. For wound dressings, also we like to use nanofibers that their surface can be modified by some biomimetic layers of polymers, for example, fibrin with attached platelet lysate components and growth factors. And we are able to prepare fibrin of different thickness, and this fibrin contains different amount of these bioactive molecules. And then we culture keratinocytes, lines of or primary keratinocytes, and try to create like stratified layer on the top. So it is interesting that it is possible to combine more molecules bioactive, like we have politely lysates here, and in next slide we have combination with growth factors, and some cells prefer different combinations. So this is with VEGF, but we have also more other combinations. And we use co-cultures here, for example, co-culture or even tree culture of 3 cell types together is possible. And then we study differentiation of these cells, growth, metabolic activity, also tightness of epithelial layer, migration of cells, production of the differentiation, expression of specific proteins by PCR. For bone tissue engineering, we need some carriers or scaffolds. They are like stiffer. They are similar to bone, so they are many times, so for example, some metals or alloys or ceramic with pores or without, but better with pores. And we see mostly some stem cells or also osteoblasts in co-culture with endothelial cells and try to vascularize them. This will be the one topic. Here's just example of which samples we had. They were 3D printed samples. It was from titanium, aluminum, vanadium alloy of different morphology or shape. And this morphology of the scaffold influence cell behavior and speed of the differentiation and creation of calcium in the layer. Here are diamonds that will be like part of this PhD project. We have more samples or projects about the diamond layers. Diamond is very interesting material. It creates some nanostructures on the surface of other metals, and cells or stem cells respond to these layers depending on the structure, concentration, morphology, or other modification, maybe chemical modification of these layers. Here we compare to stem cells type, how they respond to laser deposited diamond layers. And we see that here bone marrow MSCs were faster to differentiate and deposit calcium than stem cells isolated from adipose tissue. There is example of this reactor where diamond layers are deposited, and some by IFM some nanostructure topography of the layer, and this termination, hydrogen or oxygen termination, prepared later on this layer chemically modified the response of the cells. So it is like a typical example how the cells respond differently. Here are some images of boron doped nanodiamond. This boron has some electrical properties, and this stimulates cells, and they prefer this, and it is very important because then these processes are also integrated faster in human body, and patients are healed better and faster. These similar materials with boron may be prepared in different study. This concentration of this active, like electrically active component is very important. So we try to find out the best one. And then also in projects, we have then subsequent in vivo experiments when they are tested like implants in bones. So we now offer some diploma thesis. It is about biomimetic nanofibers dressings for chronic wound healing. This involves like nanofibers modified with bioactive molecules and their interactions with cells. And also, we will use mono and co-cultures 2D and 3 cultures, and we are going to develop a model of chronic wound. And there is some technique that they are normally used within projects. So this is one project. And the second is of PhD study project about the topic of enhancement of vascularized bone tissue using functionalized diamond scaffolds and electric stimulation. So this is interdisciplinary project when the candidate will also measure some properties of the diamond and will take part sometimes in preparation, but also they have other workers, they will do that. And in biological parts, we will evaluate interaction of cells with these materials. And also in molecular level, cellular level, so it will be more complex. So we will seek, we will want to develop vascularized prostheses that can be used later for patients. So thank you for your attention. You are welcome to visit our lab, and we can show you everything you are interested in.
Thank you, Elena. And our next presentation will be of Laboratory of Cellular Neurophysiology by two representants, Tereza Smejkalova and Aleš Balik.
Thank you. Good morning. So my name is Tereza Smejkalova, and together with my colleague here, Aleš Balik, we are looking for students to contribute to projects studying neurodevelopmental and neuropsychiatric disorders that are associated with variants in NMDA receptor genes. So another receptor. I don't know how many of you know what NMDA receptors are. So to give you a little bit of context, everything the brain does depends on synaptic transmission. The amazing abilities of the brain to receive information, interpret information, act on information, and store information, all of this depends on synaptic transmission. Synaptic transmission, of course, you know, is communication between neurons through tiny points of contact called synapses. And what happens at a synapse, the very important fundamental principle, is that the electrical signal incoming in the presynaptic cell gets converted into a chemical signal, which is the release of neurotransmitter. And then this chemical signal is converted again into electrical signal in the postsynaptic neuron through the function of postsynaptic neurotransmitter receptors. The most common synapse in the brain uses glutamate as the chemical messenger, and the postsynaptic neurotransmitter receptors then are glutamate receptors, and the two main types are AMPA receptors and NMDA receptors. So the NMDA receptors is the receptor that we know and love, and these receptors are ion channels. They are built out of 4 subunits, and these subunits surround a pore through the cell membrane, such that when the receptor is activated by binding of its agonist glutamate and its coagonist glycine or D-serine, the spore widens and the channel opens, and this is how the chemical signal turns into an electrical signal of ions passing through this channel. Most of us in the lab are electrophysiologists, so we measure these electric currents through NMDA receptor channels. The NMDA receptor subunits are encoded by genes, NMDA receptor genes that are called GRIN genes, and these are highly intolerant of variation in healthy humans. But there are variants in these genes that have been found in usually patients with some neurological, neuropsychiatric conditions, which are altogether called GRIN disorders. These variants can be missense variants that encode a different amino acid in the protein sequence of the subunit, or they can be nonsense or frameshift variants that introduce a premature stop codon, so a truncation of the subunit. Most of the pathogenic variants, so those that have been found in patients, tend to cluster in specific regions of the subunit protein sequence. In the picture, they're marked red, and you can see that both in the linear image of the sequence and also in the structure on the right, where the red dots indicate individual variants that have been found in different patients. And the pathogenic variants tend to cluster in regions of the receptor that are most important for this gating function, so for the binding of glutamate and opening of the transmembrane channel. The important thing to mention, of course, the clinical features of these GRIN gene-associated disorders are a wide range, but they include developmental delays and intellectual disability in most cases, sometimes together with seizures. Sometimes patients have more of a neuropsychiatric condition like schizophrenia or autism. There are also sleep disorders, sensory impairments, motor symptoms, all kinds of symptoms. And in many cases, these symptoms can be similar for different variants, but some patterns are beginning to emerge, such that variants in the GRIN2A gene tend to be more associated with epilepsy and schizophrenia, and variants in GRIN2B are associated typically with intellectual disability or autism. Also interesting is that the nonsense or frameshift variants tend to be associated with milder symptoms than missense variants for reasons we don't quite understand. So with this,
Okay, thank you. So I will continue more in details about our current project. So as you can see on the left side, so it's taken from the website of the patient organization Green Europe, which was established by the families of affected persons, and they try to interconnect the families, clinicians, and the scientists to more stimulate the research about these green disorders. And as you see on the map, so there's missing our country. So we were stimulated to search in Czech how many patients is here and to try to more work on this variation if there are any. So we start collaborate with the many pediatric departments in the Czech Republic in order to find these variants. And then our plan, or we are now currently analyzing some of these variants to understand the impact and possibly find the mechanism how these variants affect the brain function or neuronal function. And all these might help back to clinicians to find a better personal therapy for this very difficult, almost not able to treat disorders. So currently, in this moment, we have 9. I could say now we have already more, but so far we have 9 variants, 1 green, 3 2A, and 5 green 2B variants. 7 of them are missense, and 2 make the deletions within the gene. So none of these variants were so far characterized, so we are now very interested to deeply characterize each of these variants. As you can see on this model of NMDA receptor, so all these variants are found in different parts of the molecule, and most of them are found in the transmembrane domain, especially 2B variants, which are cause these developmental diseases. So we also have 2 variants, one which change the length of the protein. So one make them shorter on CTG side, the other change the regulatory and terminal domain. In this slide, I would like to document how we study this type of the variants in green genes. So in our previous work, we work on cohorts of the Czech patients suffering from schizophrenia, and schizophrenia is also the severe neuropsychiatric disorders, and it's one of the hypotheses is based on the glutamatergic hypofunction, so, and which is caused by the alternation in green genes. So we found many variation in green genes, mostly in the non-coding part of the gene, but also we found 4 variants in green 2A. All these variants were in the CTD region of the molecule, which is very important for regulation of trafficking and synaptic localization of the NMDA receptor. So what we have found and seen in the middle, when you express these variants in the neurons, you can see that you have less NMDA receptor on the spines, which indicate for the lower or decrease the function of glutamatergic excitatory signaling. For one of the variants, we also found that is less associated with PSD95 protein, which is very important for the holding the NMDA receptor on the postsynaptic membrane on the right place to be signal transmitted from the presynaptic side. So we study in more detail this variation, and what we have found that compared to wild type, which is in the electron microscopy figures on the left in the blue square, that the black dots are represent individual NMDA receptor. In the wild type, these receptors are in the more center of the synapse, and there are more of these dots or receptors compared to these variants with the change in the CTD part of molecule. The same we have seen on the when we analyze the synaptic charge transfer over this synapse in the neurons. So, and we propose the mechanism when these variants make less NMDA receptor on the synapse, and it's caused the hypofunction, which is in the line with the hypothesis how one of the hypotheses how schizophrenia could emerge. So this is just the flavor how we are studying, and we also use the other techniques like omics technique, mRNA expression, or protein analysis to understand the function of these variants. And together, it should bring us all information which can help the clinician to cure the patient with these variants. So and in our laboratory, so we offer the position for all stage of the study, mainly now for this PhD. So, and as I mentioned, so we do the multidisciplinary study of the neurodevelopmental and psychiatric disorders associated with NMDA receptor gene. And then there's a list of our senior members, and we have like 6 PhD students currently, and also we collaborate with many places across the Czech Republic, Europe. Also, we have many international collaboration in Europe and also in US or Canada. That's all. Thank you very much.
Thank you, Aleš and Teresa. And now there will be a time for coffee break. I think you need a little bit rest of your brain synapses. And the coffee break will be next in the next room. And for those that are connected online, we will be back at 11. Okay, let's start the second part of this morning session. And our next presentation will be of the Laboratory of Adipose Tissue Biology. And I welcome here Petr Zouhar from this part.
Thank you for introduction. So I would like briefly introduce the topic which are solved in our laboratory. We have 2 semi-autonomous units, one of them called energy metabolism unit and another one glucose homeostasis unit. We are quite diverse team, international and intercontinental right now. We have students, PhD students mainly, and postdocs from 3 continents. And we are involved in supervising students at all stages of their study career. In particular, these 3 people plus me have nowadays a PhD project advertised on the web page of the institute. But you are always welcome to contact us, even if you are interested to work on some bachelor or master thesis under our supervision. The pre-graduate students we mostly recruit from universities in Central Bohemian region or around Prague. But with PhD students, as you see, we are open to recruit people from all over the world. These are the 4 projects advertised right now as a PhD projects. As you can see, 3 of them are actually dealing somehow with non-shivering thermogenesis, and the 3rd one or the 4th one is about fatty liver disease or metabolic dysfunction associated steatotic liver disease. We are department or laboratory of adipose tissue biology, so we are mostly interested in condition of obesity and associated pathologies, including insulin resistance, type 2 diabetes, and also the fatty liver disease. So as you can imagine, the primary trigger for obesity and its consequences is some imbalance between energy intake and energy expenditure. In recent years, there was a boom of these incretin-based therapies targeting mostly on lowering of energy intake. There was so far rather limited success in potentiating such treatment by increasing energy expenditure at the same time. I mentioned that we are very much interested in non-shivering thermogenesis, and why is that? Because thermogenesis in general can represent a crucial part of total energy expenditure of rodent model and quite significant part of total energy expenditure in humans as well. There are several ways of non-shivering thermogenesis, but the problem with modeling this in mouse or in rodent model is that while human population is quite genetically diverse, when you study it in some murine model, you are usually focused on some inbred strain with very limited genetic variability. So all of the mice are genetically almost uniform. So what we tried to do slightly different was that we started with studying two different murine strains which differs in their propensity to obesity, and we plan to expand this palette of murine strains further to include some more of them. But already on these 2 strains, AJ obesity resistant and Black 6 obesity prone mice, we discovered that they actually differ quite a lot in the means how they keep themselves warm. The more studied in general, Black 6 mice rely to much greater degree on brown adipose tissue, whereas the obesity resistant AJ mice activate to bigger extent some alternative mechanisms of non-shivering thermogenesis, particularly in skeletal muscle. The typical experiment in our energy metabolism units look like this. It's based on rodent studies, so we have various murine strains or some genetically modified mice with knockout in uncoupling protein 1, for instance. Then we apply several either environmental stimuli like cold or obesogenic feeding or some pharmacological intervention, or we can manipulate the gene expression of some particular genes using adeno-associated viruses. We are also interested in how the events happening around the birth are programming the pup for later life, for later propensity to obesity. And eventually, in the end of the experiments, we have a broad palette of techniques how to monitor what actually happened in the animal, in the organism, using in vivo techniques such as indirect calorimetry, positron emission tomography, and many others. Also post-mortem analysis, including several multi-omic approaches. And we can also prepare primary culture of murine adipocytes. We are working now on development of primary muscle cell culture too. So there are several projects related to this area. I would advise you to contact us in case you are somehow interested in this topic. Probably the The hottest PhD project right now at the moment is the fourth one, which is supervised by Olga Horáková from Glucose Metabolism Unit, and it is because it just got funding this year, so there are now 3 years to reach some interesting results and to publish them, and they are really in a need for recruitment of a new PhD student for this project. So it is the project is studying again in murine, in mouse model, fatty liver disease. So accumulation of fat in the liver can lead to some steatotic liver, which could then trigger some pathological changes, including inflammation and fibrosis. So you can progress down again, farther down to steatohepatitis and eventually even non-alcoholic cirrhosis. This is triggered by imbalanced diet mostly, but it seems that quite important role in this progression of the disease could be played also by intestine or digestive apparatus, the Glucose Metabolism Unit recently established a mouse model for this progression of fatty liver disease, because it was not that easy to mimic these human conditions in mouse. And now they are on this established model. They are trying to apply several treatments to move back to healthy liver state. And one of the approaches they are trying is supplementing the diet with omega-3 polyunsaturated fatty acids, which was shown to be quite helpful in many ways. What they discovered recently is that also the form in which omega-3 polyunsaturated fatty acids are administered to the mice matters a lot. So phospholipids are much more potent than triglycerides to change anything. They are also studying the mechanism, molecular mechanism, what's happening in the liver if the during the progression of the disease and during the regression. Also, they are interested in regulation of autophagy. Also, what's happening with the peroxisomal function, and most recently, they are getting deeper into the barrier function of gut and mesenteric fat in protecting liver against the deleterious effects of imbalanced feeding. So that's all from my side for now. It was just a very brief overview. If any one of you is interested in further details, I'll be available for discussion in the afternoon.
Thank you, Petr. And the next presentation will be Laboratory of Molecular Physiology of Bone, and I welcome here Misha Tencerova.
Thank you, Olga, for introduction, and also hello, everyone. Just continuing on the topic of different focus on different organ systems. So we actually study bone homeostasis in connection of metabolic disease, as you heard from previous presentation, that actually obesity causes different metabolic complication, and the bone is also affected by accumulation of fat also in this organ, that it's associated with the higher osteoporotic and fracture risk. And that there is the aspect of the sex dimorphism, that also the literature gap, and also the future strategy in the treatment and efficacy of the treatment. It's important to consider also the effect on the men and women, because our system and our physiology is set up differently. And also it's the same for a bone. And in terms of the pathophysiology in the bone system, we know that for osteoporosis and the fracture risk between men and women, it's already a difference in terms of the prevalence. 1 to 3 women and 1 to 5 men actually suffer from the fracture complication after age of 50, and that there is a huge impact on the homeostasis inside of the bone marrow, because we have this higher impact of the sex hormones, and also the aging effect that affects our body differently. And that in the side of this bone marrow we have these stroma cells that they are contributing to these function and the bone homeostasis of our skeletal system, it's very important to know why this pathology is differently affecting men and women. So from the point of animal studies that we apply in our labs for animal models of obesity, usually apply with high-fat diet model that you also heard from the other presentation, but also in terms of the model for postmenopausal osteoporosis with removal of the sex organs such ovaries in females that can induce actually the complication in the bone homeostasis. When we look actually just by high-fat diet between male and female mice, the impact on the glucose tolerance is differently present, and also with the weight gain with response to the high-fat diet. However, here on this picture, you can see that the adiposity inside of the bone marrow between male and female mice is differently presented and accumulated. So this is also some of the other aspect that we would like to study. That what is the actually molecular mechanism between the setup of the skeletal system between male and female in a condition when we treat them with different diet composition. So we also perform the molecular analysis of the skeletal stem cells of male and female mice on normal and high-fat diet condition. And we got some interesting targets that we would like to follow up on this in terms of their prevalence, in terms of differentiation potential, and also analyze on their metabolic pathway. So from this, we would like for the PhD or a master project, depends on the deep analyze what we can actually focus in specific aims, identify the metabolic pathways that contribute to this obesity-induced phenotype inside of the skeletal stem cells with respect to the sex dimorphism, and then to look at with different condition, with different diet composition, exercise, or with removal of these sex hormones, how it impacts actually the bone and full body metabolism. How we actually study this. So just a brief overview of different methods that we use in our lab for in vivo characterization of the bone and fat metabolism. We are using the characterization of the metabolic complication during the diet. Then also the bone phenotype by scanning by a DEXA scan or micro CT, where you can see also different structure of the bone characteristics. And we also are interested in bone marrow adipocytes, as I mentioned. So using different contrast agents that we also collaborate with the lab in Belgium, that we can actually, by reconstructing of the images from the micro CT, to look at the distribution of these different compartments inside of the bone, because it's not so easy to see through the bone or core of the bones, that this distribution of these adipocytes inside of the bones and the interaction with the trabecular and cortical bone parameters is important for our bone strength and the quality. That it's the impact that we are also studying in the context of the sex dimorphism, but also in terms of different interventional treatments. So then you can really visualize these nice structures of the bones and the adipocytes or the vessels, which takes also some image analysis from the micro CT. Other ex vivo analysis that we perform is by histology, that you can actually nicely visualize different cell types and how they actually interact with different compartments, from osteoblast, osteoclast, or collagen analyze, and also the testing of the quality. And then the perspective of isolation of the stroma cells. We are also isolating these primary cells from mice, or also some clinical samples, where we can, after isolation and adherence to plastic, perform several assays to measure their metabolic assays, their response to different stimuli by molecular analysis of their different pathways they are involved and affecting their function inside of the bones. So if you are interested also today we have active days, we have the dissection. So that's a lot of going on in our lab. So if you would like to also see like, the lab work in action, so you can also come in the afternoon. And also if you would be interested in some other topics, so you can also contact me or visit our website. Thank you.
Thank you, Misha, very much. And next speaker will be Onsei Kuda. He's going to register his presentation, and he's from a laboratory of metabolism of bioactive lipids. And please, Ondra.
Yeah. Thank you very much. Good morning. My name is Ondrej Kuda, and yes, this is my lab, metabolism of bioactive lipids. So we study lipids. These lipids should be bioactive, meaning they have some role in life. And how we do it is this. You are all interested in physiology, and I hope you know this omics cascade. Basically, we start with DNA through RNA, protein, metabolite, flux to the phenotype to the life. So my lab is interested in this part, the lower part. Basically, what we are trying to do, we are trying to integrate all the different omics. We usually start with transcriptomics and then proteomics. We do ourselves the metabolomics and lipidomics, and we are trying to integrate all of these together, including fluxomics, to get something out of it and to find metabolic regulations in health and disease, different modals. So we basically check how metabolite pools do something, and how they do it means what happens at what metabolic rate, the enzymes involved in these reactions, and then how this impacts all the living organisms. So my lab focuses on metabolomics, lipidomics, and fluxomics, and we use also these other omics. As an example of such omics integration, I'm showing you a recent article from our lab. My colleague Misha Tencerová already introduced the bone and bone marrow. So here we use these three panels, metabolomics, lipidomics, fluxomics, and some models, the phenotype. And here I just briefly describe what's going on. There are pills called rosiglitazone and pioglitazone, and these pills are for diabetic people, and these compounds help these diabetic people to control glucose levels. That's good. Unfortunately, these drugs have side effects, and one of these side effects is adipose tissue within the bone, and too much adipose tissue within the bone makes the bone fragile. So Misha already introduced this. And then we were interested in this new compound, which is kind of derived from these older two. This is a newer one. It is called MSDC 602. And this compound is somehow different because it is able to prevent the health of the bone. And that's good, but no one knew how it works. So here we integrated all the omics data available, and we used metabolic flux analysis, meaning we measured the rates of metabolic reactions using metabolites or tracers labeled with stable isotopes. In this case, it was 13 carbon. And we found that there is some kind of rewiring of metabolic pathways, which is beneficial in case of this new drug, but is not that beneficial in case of these old drugs, pioglitazone and rosiglitazone, and this is the reason for the side effects. All these three drugs share the same mode of action, which is inhibition of mitochondrial pyruvate carrier, this one, which basically shuttles pyruvate coming from glucose into the mitochondria to generate energy. And we had to show that there is something going on at this step. And to do so, we created new subcellular probes, meaning chemicals which can label metabolites within the cytosol and within the mitochondria separately. And we were able to show that there is a difference between these two compounds and this one compounds, how they operate, how they modify the metabolism of the mesenchymal stem cells within the bone marrow. So this is just for you to see what we are working on. And we have some PhD projects. The first one is a project in biochemistry. We would like to decipher metabolism in cancer. I mentioned that we are interested in health and disease, and this work is focused on cancer, specifically cancer cachexia, which means the wasting condition. And this project focuses on mouse physiology, a lot of mouse surgery, also metabolic fluxes, as I showed you before, and then a little bit of bioinformatics. So this is basically a lot of biochemistry. The second PhD project is focused on bioinformatics. So it is something different. This is the integration of metabolomics and lipidomics, as I showed you before. So a lot of numbers, a lot of coding in Python, R, or whatever you like. It really doesn't matter. But it is focused on the metabolic calculations. We have also some undergrad projects. One of them within the animal physiology. We would like to optimize the catheterization and infusion protocols in mice, meaning mouse surgery, insertion of catheters, infusions, work with animals, a lot of physiology. And another option is more bioinformatics, and these are very small projects like coding in Python, MATLAB, C# or Bash, small applications or software tools which help us to work with the omics data. We need a lot of normalization, standardization, harmonization in the field. So this is focused on LC-MS-MS, liquid chromatography mass spectrometry, or GC-MS-MS method, work with the raw files, machine learning, and a lot of other bioinformatic tools. Yeah. So if you are interested in anything of this, just meet me at 1 p.m. here, and I can show you the labs and people. Yeah, oh, sure. So thank you.
Thank you very much, Ondra. And now it will be my turn of our laboratory of membrane transport. Okay, so welcome to our laboratory of membrane transport. I am a head of this laboratory, and I am offering a bachelor or master science thesis. And as you heard in the introduction, the research at our institute includes research from the molecular level up to the whole organism. And our laboratory is really at the bottom of this pyramid because we study cell membranes. Cell membranes are selectively permeable, and to communicate with the environment, cells needs transport, and which is crucial for life. And we study transporters, particular membrane proteins that transport compounds and signals across these membranes. And they are really necessary for the uptake of nutrients, efflux of waste compounds from cells, for the communication with the environment, and particularly we are interested in those transporters that are crucial for ion homeostasis. And we are a unique laboratory which uses as a model organism of an eukaryotic cell yeast Saccharomyces cerevisiae, which is not useful only for the production of beers and wines and bread, but it is widely used also in research as a model organism of eukaryotic cells because we can use this yeast as to discover molecular aspects of various fundamental cellular processes that are highly similar to high eukaryotes. The advantage of using yeast is the rapid and cheap cultivation. And we have enhanced highly developed methods of molecular biology and genome engineering. And we are, as you will see later, we are using yeast also for the study of proteins from higher eukaryotes and to develop something like humanized yeast, a simplified cellular model of human disease. There is a list of methods that we use in our lab, ranging from bioinformatics, protein modeling, structure analysis, molecular biology methods, biochemistry, microbiology, and also physiology of cells, as well as some biophysical methods. And we collaborate also within our institute with other departments concerning the structure of proteins, and also in the we have a collaboration within the campus, particularly as concerned using advanced optical microscopic techniques, and also we have some collaborations abroad. We are a very small group currently because our previous boss retired, and in January I became head of this laboratory. So we have two researchers, Clara and Misha, who is now she is now at the maternity leave, but she will come next year. Pavla, our technician, and we have bachelor student Mikhailo and Christina. And for a long time we participate in the project of Academy of Sciences, Open Science, and currently we have two students from the secondary school, Ella and Veronica, and there is a place for, as I said, for the master science or bachelor student, and you are very, very welcome. And the project that we offer has a general title, Cations, Key Elements for Life of Eukaryotic Cells. Because for any living cells, there are monovalent cations crucial, like protons or hydrogen cations. The concentration determine pH and also electrochemical gradients across the membranes, and as well as sodium and potassium cations are necessary for our lives. And in general, from bacteria to animal cells, there is a dogma that potassium is highly accumulated in cells and sodium must be eliminated from cells. And any disturbance of this homeostasis results in various pathologies at the cellular level up to the whole organism. And for this, it is necessary to have a tight regulation of transporters. The membrane proteins that you can see here at this picture, the schematic proteins that have various transport mechanism, various type of transporters. And various with different substrate specificity and transport mechanism. And what we are interested in particularly, we studied the structure and functions of cation transporters from yeast. Many of them that you can see at this model of yeast, and that they're in this pictures. We characterize them and their structure and substrate specificity, and we studied the first fungi-specific transporters like this potassium importer, TRK1, because currently maybe you know that there is a big problem of high resistance of yeast cells to antifungal drugs. So we now would like to search for possible new antifungal targets, and one of them is this TRK1 transporters, because it is present in yeast cells but not in human cells. Then the next topic that we are interested in is the organization of transporters within the plasma membrane, because there is very few information about the organization of particular proteins in the plasma membrane and how they change their localization within the membrane, for example, in response to various conditions in the extracellular environment. Here you can see the microscopic pictures of transporters that are targeted or tagged with various fluorescent probes, the green or red one, and we studied the relation between them. We are also interested in biogenesis of these complex membrane proteins. They have usually highly complex structure, these cation transporters. And you can see here an example that when we remove one very small protein which is present in endoplasmic reticulum, and the protein which should be in the plasma membrane is suddenly stuck in the endoplasmic reticulum, and so this small protein is highly important for the biogenesis of this membrane protein. And we also try to discover new regulatory mechanisms of transporters at the molecular level, like and find out some regulatory proteins. Like here you can see an example of our recent studies that 14-free regulatory proteins are involved in the regulation of two of the transporters that we characterized. And as I said, we also use yeast cells for the characterization and structural studies of human transporters. Recently, we discovered many new information about one human NH sodium proton antiporter. We also analyzed the inhibitor binding site in this protein. And currently, the most important for us are also to try to express potassium channels. So if you like to know more about these topics, you are really welcome into my lab and visit us in the afternoon, or for those that are online, you are really welcome to write me an email. Thank you very much. And I will welcome here the next speaker, who is Martin Balaštík, and from the Molecular Neurobiology Lab.
Okay, thank you very much. And hello, everybody. It's really my great pleasure to be able to introduce to you the Laboratory of Molecular Neurobiology that I am head of, tell you what we are interested in, what kind of methods we use, what is our focus, and why is it actually all important. Now, if you know that, human brain, you may know that it represents only 1 to 2% of the mass of your body. But actually, this is the 1 or 2 critical percents that makes us human. And we know that it's not the mass of the brain that we are unique among all the species. It is what is actually inside the brain, neurons, glia, and it's not the number of neurons that we would would be unique and have the highest number of neurons among the species. It is their connectivity. We know that with the 10 to 14 of synapses in the human brain, currently there is no other species that would have more synapses or connections. And we think that this is related to our unique cognitive functions of the humans. Obviously, the sheer number of synapses and connections within the human brain makes it very difficult to analyze and study. And we know the connectivity within the human brain more on the level of the regions than the synapses themselves. As you may know that recently the full connectomics on the synaptic level was a huge achievement published and finished for Drosophila brain, which obviously has much fewer number of neurons, only 140,000, forming 50 million synapses. Now, in my laboratory, we are not interested that much about analyzing the connections within the brain, but more as molecular biologists. We are studying the mechanisms and genes that are critical for formation of the connectivity within the brain. And we use animal models and study, particularly as for in the region of developing cortex, various milestones and steps that lead to formation of cortex, which is the proliferation of the progenitors, their neurogenesis, formation of neuroblasts, their migration and formation of the cortical layers, and then their differentiation, the full differentiation, and formation of connectivity. Now, all these steps are extremely well regulated in mice as well as in human. And we know that changes in the proliferation, neurogenesis, etc., may lead to various neurodevelopmental disorders, like autism spectrum disorders, or epilepsy, or schizophrenia. And while proliferation or migration are very different, they have one thing in common. They are mechanical processes, and they need some motor system that is just pushing it through. And this is obviously cytoskeleton. And we know now that particularly microtubules play a very important role in formation of the cortex and the axonal growth, as well as the connectivity formation. And this is already a subject of our interest in my lab, where we study how different regulations or deregulations of microtubules affect axonal growth and brain formation and on various levels. So we are focusing on post-translational modifications of microtubules, be it polyglutamylation, detyrosination, phosphorylation, how they affect the microtubule properties and the development of the cortex. Various microtubule-associated proteins. We know that they are binding and they are affected to microtubules, and they are affecting the activity or function of microtubules. And obviously, they are also post-translational modified, phosphorylated. We are testing how changes of the post-translational modifications or their conformation changes, how they affect the brain development. Finally, we have now a new project, new direction that I'm actually more advertising here, that is related to the adaptor proteins like TRAC1, TRAC2, that are linking motor proteins, kinesin or dyneins, anterograde or retrograde motor proteins, to mitochondria, and that are actually they have very important role in distribution of mitochondria within axons, and they actually have a critical role in the development of the brain connectivity. So basically, we are testing all these different areas, or these different aspects of microtubule regulation, and test how they affect axon guidance, synapse formation and refinement, and brain development in the mice models, and various neurodevelopmental disorders like autism spectrum disorder, malformations of cortical development, epilepsy, etc. Now, I will just use the remaining time just to describe some methods that we use to achieve, to reach these goals and to address the questions that we raise. First, we generate and analyze knockout mice. We have generated already several knockout mice and analyze their phenotype in great detail. We use TALEN's mutagenesis, CRISPRs, and we were successful to generate even isoform-specific knockout mice. So then we are modifying or labeling the developing neurons in the cortex using in utero electroporation, which is a rather unique method that allows to label some specific part and specific number of neurons in the developing cortex already inside the embryos, and then basically ended up with neurons in the cortex labeled before they fully developed and differentiate. And this is what you see here. These are the in utero electroporated neurons that were labeled by GFP expression, and then they basically, we can trace their projections both as for the dendrites and analyze them in the synaptic level to test how many synapses are formed there, as well as in the contralateral cortex to test their connectivity with the neurons in the cortex, in the contralateral cortex in the other hemisphere. We are using the slice cultures of organotypic brain cultures to test the migration of neurons that we labeled by GFP using the in utero electrophorations. Here, basically, we are able to label the currently generated neurons and trace them, how they migrate within the cortex, and then basically analyze their trajectory as well as the distance, etc., which gives us a very good idea about how various mutations affect formation of layers within cortex. We use gene gun labeling of either plasmids or some fluorescent dyes that allowed us to, within cortical slices, to randomly label neurons in their entirety and then basically analyze their connectivity, number of synapses, their morphology, etc., which is something that is, again, very useful to analyze the effect of various gene mutations. Here is actually the number of synapses, the dendritic spines within the mutant mice that we analyzed. We use and generate many primary neuron cultures and explant cultures, again, to use together with immunolabeling to analyze the effect of various gene variants and mutations on the connectivity, on the axonal growth or connectivity formation. And we do it also in the microfluidic chambers, where we separate dendrites and axons, and basically you can analyze specifically axonal growth as well as somehow affect it and treat it only the axonal part. Which is also very useful in our studies. And also we, in collaboration, particularly with the Institute of Biotechnology, we are analyzing and testing the effect of various microtubule-associated proteins or the adaptor proteins in vitro, where we can really, on in vitro grown microtubules, test the effect of, here, for instance, CREM2, microtubule-associated proteins on microtubule polymerization. And this really gives us a very good idea about the mechanism, how these microtubule-associated proteins work, and how they generate the phenotype we analyze in mice. So together, we are kind of trying to generate a map and to analyze and characterize the microtubule regulation on different levels, from single molecule in vitro to cellular or neuronal level in neuronal cultures, through systemic impact in various mouse models, and through our collaborations in hospital, the major hospitals here in Prague. We are actually linking it to various neurodevelopmental disorders in human patients. Okay, these are some of our recent publications on this topic, and I have to thank particularly my lab members that are great, and they are just they are able to do excellent work and analyze the mice that we are generating, and our collaborators, of course. And if you are interested in any of these subjects, I would be very happy to talk to you. We are now opening a new project with regards to the adaptor proteins, their role of mitochondria, transport, energy metabolism inside neurons, and the effect on brain development in vivo. So you can write me, you can talk to me. Unfortunately, today in the afternoon, I'm not going to be here. So, but you can still visit our lab, talk to the students there. But you can definitely write me. And for this moment, thank you very much.
Thank you very much, Martin. And the last presentation of the topic that we offer is from the Laboratory of Experimental Hypertension. And I welcome here Honzanecka.
Thank you, děkuju. Dear colleagues, because lunch is coming, I will be, I hope, brief. And let me introduce our Laboratory of Experimental Hypertension. As you know, hypertension is a general problem in the world, especially in the West world. And this illness has a very specific effect for many biological or organ system. It's you know very well that it could affect atherosclerosis. It can induce it can be responsible for brain ischemia, which is the reason for stroke, also for some retinopathies. And the main information probably we will know about the renal failure and chronic heart failure. And these two parts are also in the center of interest in our lab. So here you can see some members of our team and topics which we are studying. Therefore, in the center, there is high blood pressure. And our colleague, Dr. Zicha, and together with Dr. Rezáčová, they are studying the central and peripheral mechanism of blood pressure controls. Head of our department, Dr. Vaničková, together with Dr. Hojna, they are studying the effect of glyphosines in non-diabetic hypertension and chronic kidney disease and heart failure. And I a little bit mention about it later. Dr. Kunesh, he's working on the project dealing with neuropeptides in obesity together with chemists from Institute of Organic Chemistry, Academy of Sciences. There is also a small group, Dr. Karouchova and Kadlecová, they are analyzed oxidative stress and role mitochondria in many cardiovascular diseases. And finally, subgroup or people which are interested about the autonomic nerve system in blood pressure regulation and chronic kidney disease and heart failure. And in this part, I am working together with my PhD student, Almos Boros. And now I would like to mention something about our experimental models. We are working mainly with rats, and there are some subgroup of genetic models, genetic modified rat strains. We are working with spontaneously hypertensive rats. These animals are characterized that they are develop spontaneously high blood pressure. Another rats or strain rat is dole-sol sensitive animals. These rats, if they are treated by a high salt, means high level of NaCl, they developed again a very severe form of hypertension, and also some genetic model ran to transgenic rats. In this case, these rats has a mouse gene for mouse renin, and probably you know that renin-angiotensin system is very important for regulation of blood pressure. But also we are used some experimental model which were hypertension is induced by, for example, reduction of renal mass or renovascular hypertension, which is induced by model 2 kidney 1 clamp model. On this slide, I would like to show you some our model which has effect on heart, because heart is the main part which is interesting for me. So if we have our animal model, and if we treated rats by high-fat diet, you have develop obesity, and after that we are analyzed these rats. Another model which we use it for heart failure is model of aortic constriction. When you can see here, there is a sham-operated heart. Normal size is about 1 centimeters in diameter. And here you can see in the same magnification hearts after aorta constriction binding. On this part, you can see heart after myocardial infarction. It's a cross-section area of heart where this part was infarcted. You can see very thin layer of left ventricle, left free wall, and hypertrophic part, especially in the septum and free wall. We are able the function of myocardium to detect by echocardiography, and there are two, maybe it will be not so good visible, but okay. There is normal heart, and this is the size of chamber in a normal sham-operated heart. And here you can see very thin layer of walls and dilation of left ventricle. So if I can summarize this part, in our department we use a lot of methodological approaches which go from all animal measurements, as I mentioned here, radiotelemetric measurements or monitoring of blood pressure and heart rate, direct measurement of blood pressure, etc. We also analyzed organs like perfused vascular bed or isolated organs. Also, we used cardiological approaches, echocardiography, and also catheterization for analyzing of pressure volume curves, and which is everywhere in each lab in institute. We also used immunohistochemistry methods, biochemistry methods, or molecular biological methods. And about the project, which could be, which you can be also involved there, we have there 3 projects. Let me tell something briefly about these topics. The first dealing the role of hypoxia-inducible factor 1 alpha, the progression of heart failure with preserved ejection fraction. Hypoxia-inducible factor 1 alpha, it's a transcription factor, which is like a master which regulated oxygen deprivation in heart and in cells. And our recent experiments showed us very interesting findings. We used controls pregoldy rats and rats which were genetically modified. It's because we have a very nice collaboration with the department of Professor Dr. Pravenec in our institute, and they created for us these Sprague-Dawley normotensive rats, which has a heterozygous expression of HIF1 alpha, this hypoxic inducible factor alpha. And we treated these animals by a high-fat diet, LNA, and together with LNAME is an inhibitor of an all synthase. Therefore, we developed something like a model of Western diet or Western lifestyle. And as you can see, there is a survival rate in our experiments. After 10 weeks of treatment by high-fat diet and LNAME, we observed in heterozygous HIF1 alpha rats higher mortality compared to SD rats. Therefore, rats which has a normal HIF1 alpha level, there was no difference in mortality in controls. And we measured a lot of interesting things, and one of them was plasma concentration of uremic toxins. Uremic toxins, it's compounds which are produced in gut in gastrointestinal tract, and normally they are eliminated by kidney. And if you have some kidney injury, after that you have a higher concentration of these compounds in plasma. And you can see that especially in HIF1 alpha heterozygous, after high-fat diet and L-NAME, we observed really high level of one of these uremic toxin with the name of picresol sulfate. The second project focused on senescence, therefore aging, in cardiovascular and renal disease in hypertensive rats. And again, I would like to demonstrate to you some our preliminary data. Here you can see spontaneously hypertensive rats with age 22 months old, and these rats we treated by compounds with name of mitotam, which has some senolytic effects. It means that they are reduced production or reduced the number of senescent cell. And here there is the hard concentration or positivity for collagen. This blue part represents collagen, and you can see in untreated rats there is a lot of blue, and rats which were treated by compounds MT, mitotam, there was strong reduction of collagen positive tissue. A very similar findings we observed also in kidney. Again, if you compare blue color on the left side and right side, you can see a strong difference. And finally, the last project, it's dealing with empagliflozin. About gliflozin, or gliflozin I mentioned at the start of my talk. And gliflozin, it's inhibitors, they are inhibitors of sodium glucose cotransporter 2. And the main function of gliflozin is glycosuria, therefore the production of or elimination of glucose from blood. But it has been shown that gliflozin has also a very positive effect for other cardiovascular parameters and renal parameters. They increase natriureases, they reduce proteinuria. They have a metabolic effect, therefore they have something effect like caloric restriction. And during last year, it has been also shown that they have a cardioprotective effect. They reduce production of reactive oxygen species in myocardium and reduce intracellular concentration of natrium and calcium. So finally, gliflozin has a renal, cardiac, and vascular. They are reduced to renal, cardiac, and vascular inflammation. They have a renal protective and cardioprotective effect. So it was the third our project. And finally, I would like to comment our collaboration at the Institute of Physiology. We have a close collaboration with Department of Development Cardiology and Genetic of Model Disease. And it's important to also mention that we have excellent collaboration with colleagues from Institute for Clinical and Experimental Medicine, especially from Experimental Medicine Center, Cardiovascular Surgery Department, and Cardiology Department, and also another faculties at Charles University faculties, which are mentioned here. And that's all. And please, if you're interested about cardiovascular topics, please contact myself or me or our head of department, Dr. Vaničková, and thank you for your attention.
Thank you very much, Honza. And I would like to now welcome here 3 real students from our institute, PhD students, Amanda, Bara, and Daniel. And they will tell you more about their experiences from our institute, so you can see in reality what you can expect at this institution. Will be the first, Daniel. Amanda.
Hello, everyone. My name is Amanda. I'm from Brazil, very far away. And I got to know this position through this website here, Find a PhD. And then after the interviews and I got accepted, I needed to apply for a visa. And then I could come here. So I am a second-year PhD student from the Animal Physiology PhD program. And I am working at the Laboratory of Cholinergic Signaling you guys heard from my supervisor, Dr. Helena. And I'm very interested in studying the function of nicotinic acetylcholine receptors in the striatum. So this striatum, it's a very nice structure in the brain. It's very important for so many behaviors, such as control of motor or motor control. So from very simple motor tasks to complex movements, and also it's very important for habits, cognition. So cognitive tasks such as decision-making, reward processing, and social interaction. And then I'm very interested in this population of neurons in the stratum called cholinergic interneurons. So particularly, my work is to study the role of nicotinic acetylcholine receptors expressed by cholinergic interneurons, this population here, in the control of striatal activity. And then this way we could explore how these cholinergic interneurons influence in these roles the striatum is quite important for. And then a bit of my interests, I really like music, dance, moves, watching movies, and also going for music jams and pub quizzes. And I also, as a PhD student, we're always trying to participate in scientific events. And also, I would like to call your attention for a research collaboration we have at the lab, and then I could spend 6 months in Brussels for this research collaboration to learn patch clamp electrophysiology. So this way, I could learn how to measure the activities of those cool cholinergic interneurons. And then apart from the scientific part, I also got to know the university or enjoy there. Also know Brussels, which I had never been before. And then also explore the food, so the delicious waffles in Brussels, and also make friends. So now I will pass the word to my friend, Daniel.
Friends? Are we friends? I'm kidding. So, hello everyone. My name is Daniel Vasconcelos, as Amanda said. I'm Portuguese. I'm living here in Czech Republic for 3 years now, and I still don't know many Czech words, but I'm working on it slowly. So, as Amanda, I saw the advertisement. It's some international website. It can be Find a PhD or someone else or some other else. Sorry. And after getting in touch with my supervisor, we decided that I could apply for this PhD program. So I did. I'm now on my 4th year of PhD, and I'm part of the Department of Pain Research. That you already had a talk in the morning, I think. So you must know more or less what I'm talking about. And I'm studying chronic pain. So as you know, it's affecting, or you might know at this point, is affecting 10% of the worldwide population. And there's increased incidence due to the aging and survival after chemotherapy treatment to treat cancer. So there are people developing some chronic pain, and there are no good medicines to treat it. So that's why we are studying to try to understand the mechanism and how to treat. So, as a PhD student, I had the chance to travel a bit all over to present my results, which is a cool thing you can do as a PhD student, and get to know another labs, another projects, and other works, and another countries, of course. So I've been in many conferences. I have only here 3 of them. And my interests are, basic ones. So I'm kind of sportive guy. And I like music, and that's basically all. So before moving for a PhD, the thing that you should ask yourself is like, where do I want to go? So either the institute or the group that you are joining. And I did this thinking. And for me, the most important thing was like the topic I would be choosing for doing my PhD, and the supervisor, and then also the institute. It should be important. So you should consider this. Then you will have many challenges and things to overcome. And you will realize that to overcome the challenges you will have, you will need to be surrounded by either another PhD student that can help you or other people that you can ask whatever you need to solve it. So this is not a one person job, you know, you will be always surrounded by the community and you can find some help within. Then there are many researchers highlights like the results you will have. Then you will have many good moments, you know, every time something is working, you are proving your theories and so on. And my advice is you should really think about, you know, all these things and be aware that this is a global thing. You can be part of this, and it's important for you to be surrounded by people and discuss with everyone. Don't be afraid to do it. But I'm pretty much losing my time thinking about this because I do surf and I'm always looking forward to go back to Portugal and, you know, go there and surf. So now I will pass. Talk.
Thank you. Is it working? Yeah, perfect. So, hi, my name is and I am from Public, and I will tell you my PhD story. So I am coming from Olomouc, where I did my bachelor and master. And when my master studies was coming to the end, I was thinking, what now? So as I knew that I want to continue in the science and to be specific in the academy, I was searching various pages, and then I came to very interesting topic in the webpage of the Institute of Physiology. So I apply. Like my supervisor saw that I am good candidate, so she accepted me, and I enrolled in the program of animal physiology under the Charles University. And now I am in my 4th year as a PhD student. But all the job that all the job that I do is here in the Institute of Physiology, and to be specific, in the laboratory of developmental cardiology, which is one of the biggest and for sure oldest, but like great for sure. Laboratory that focus on the cardiobiology. And here I focus on the mechanism that protects the heart. I am focused on HIF-1 alpha that you heard about it before, also in context of chronic hypoxia, and my main focus is on the mitochondria. During my PhD, I had the chance to try to live abroad, so because I went to internship in the University of Padua. There I had chance to work with lentiviruses and prepare some very cool cell lines. And also I had chance to work with very advanced microscopy. And also I was lucky to go to some conferences where I could present my data. Here is just some of them that seems to be more interesting. And in all of these conferences, I had active participation in most of them by poster. And in the first one in Seoul, I even gave the talk. So that's all from me. And I will also shortly speak about like institute activity that what we have kind of special here. Is that this institute is organizing PhD conferences every 2 years. So you have great chance to train your presenting skills and of course having a lot of fun and meet the PhD colleagues and so on. And also there is another PhD conference that is organized with Polish Institute Nensky. So you have also a chance to travel abroad and basically have the same PhD conference and again present and so on and meet people like from Poland. Then also Institute is organizing every 3 years all employee meetings, and by chance is this year, and we will go to Spitler. And the last slide, so it's almost over. Please stay with me. We have also some PhD activities in our institute. We have an organization that is called ECRA, and it's basically it's PhD students organizing activity for PhD students. So in the past, we had activities like board games, bowling, or picnic. And also some kind of this organization is in many other institutes. So There is also some another events in another institute that you can participate. So it's also very nice to find new friends and find somebody who you would like to collaborate. So that's all, and I will leave.
So just to wrap up everything, so you can be part of this shaping the future of science. If you feel like doing a PhD and choosing our institute to do the PhD, you know now that you will have the support from the other PhD students, not only from your supervisors. So you are more than welcome and, be part of the future. That's all. Thank you for attention.
Well, thank you, all of you. So you had the opportunity to hear the real stories of particular students, and that we have some also not only scientific but also social life. And I think now we can stop the morning session. Only those that are online, they will have a possibility to ask the question, or you can ask also now the questions if there are any, or we can talk outside during the lunch refreshment. So are there some questions from you now? Just to organize the afternoon session, so take the lunch, and during the lunch register to these papers for the visits of the laboratories. If you like, you can choose up to 3, or if you like to visit more, just tell me, and we can organize it somehow. Yeah. And here I see David.
Yeah.
Yeah. Hi, David.
Hi. My name is David, and I have some questions to ask.
Yes.
Specifically, I want to know about the PhD program, if it's fully funded.
PhD program, Martin.
Yeah.
Can you answer if it's fully funded? It depends on the laboratory. It depends on the grant availability at particular laboratory, and you are always the student of some university, mostly Charles University in Prague. And there you have to pass the concourse for the fellowship for PhD study. But I will pass the microphone to Martin, who is responsible for PhD study at our institute.
Okay.
Okay, hello.
Hello.
Okay, you hear me. So actually it goes in a couple of steps. There's first on our website, there is a form to apply to the PhD program. And those selected candidates will then need to go for another round of selection within the university. And those the candidates that are selected, then they are getting the stipend for 4 years plus the laboratory where you will do your PhD here at the institute is getting up to full FTE salary to the candidates or to the PhD students. So yes, it is funded. The exact number is then discussed during the interviews with the PhD supervisors and the faculty. Okay.
Okay. So the selection process is like competition basis, right?
Right.
And so those that are not selected, is there provision for self-funding?
I'm sorry, I didn't hear you.
Self-funded. If you're not selected for the funding, is there?
Yeah, I mean, the selection is just that you have to fill up your CV, your experiences, your study results. This is all on our website. So this is the first, and then usually there is a, if you are the good match for any lab that you apply to, then they will contact you and you will have a Zoom call. So I don't think that there is any cost related to that.
No, I'm not talking of the cost with the application process, but you have to go through the interview, and then the school will accept you, and then the laboratory will take you as a PhD candidate. And what I'm trying to say is that those selected, they were selected to be funded by the PhD program.
Right. I mean,
yeah. Then those that were not selected, can they self-fund their PhD study? Is there provision for self-funding students?
Oh, okay. So if you're for that I think that really entirely depends on the discussion with the PhD supervisor. I think that you can, if you are a good match and he wants to have you in the lab, maybe he can find some different ways how to find the support, or even without support if you have your own funding. So that it's all possible. I think that just the first step. This is the most important, to fill up the form that is online and select the labs that you're interested in, and then they have to, among all the candidates, then they have to pick you and discuss with you, and then that's where it starts. And this is if you finish this successfully, I think that there is a good chance that you have all.
Okay. It's okay. Thank you for your response.
Okay, thank you.
And answer.
Any other questions from online participants?
Hi, Olga. I'm Andrea.
Hi, Andrea.
Hi. I'm based in London at the moment, but I'm really interested about a PhD. And I was wondering, for who is online, there is any chance to view the labs in some way, even through videos, something like that?
Videos, not yet. We plan to make some more video presentations next year, but there is on our webpage there is some interactive presentation of some labs, but it's quite old, I would say, and it's in a more popularized form. But you can watch it. Yeah.
Okay, fantastic. We'll rely on that. Thank you so
I
much.
think nowadays it's not problem. If you are interested in some particular lab, you can contact the supervisor, and he can via WhatsApp he can show you the how the lab look like, for example.
Okay. Okay, thank you. I will consider your recommendation.
Thank you very much.
Thank you, Olga.
Any more questions from there, from and online? No, and here in the audience? No. So I think we can finish the recording, and I really thank you all of you that you attended online, and you watched now the record, and we can stop it now. Bye bye.